together. My presentation topic is finite element estimation of calcium ion in presence of NCX and buffer in astrocytes. We have taken here two parameters, that is first is the buffers and the second is the NCX, that is the sodium calcium exchanger. What is the role of calcium that we see? This is the content, that is the introduction. First, we understand what is the role of calcium ions when in our nervous system. Nerve impulse flow from one nerve cells to another cell, and the only one region is the calcium ions. As calcium ions enter into the cytosol, what happens? The nerve vessels, which is in the form of neurotransmitter, that moves towards the another nerve cells, or maybe the between the nerve cells, that is the synapse. So that as the calcium concentration goes high and high, the process happens, but at the same time it goes at the high level, it becomes a toxic for the cell. And this is the mechanism for the movement of the nerve impulse from one cell to another cell. There are so many parameters that affect the calcium concentration level. It means what? That control the system. Suppose, for example, we are talking about the calcium buffering. Buffer is what? It is a one kind of protein that we see in the next slide. Our main purpose here to study the sodium calcium exchangers. At the 2 decades before, astrocytes are not taken seriously as a member of the signaling processes. It will take they feed the neurons, whatever, so that we have considered astrocytes. And then NCX, the role of NCX is now emerging nowadays for the signaling processes. Sodium ions affect the cytosolic calcium concentration level, and naturally it affect the signaling processes. In this paper, we have taken the two-dimensional mathematical model that we have solved using the finite element estimation. Using the MATLAB, we have simulated the results. Some literature we have taken here for the just like this Smith et al. Have taken the buffering process, calcium buffering process, and they have solved using the finite difference methods. Tiwari et al. Has taken the sodium flux, and they have shown the impact of the sodium on the cytosolic calcium distribution on the neuron cells. This was my paper. Here we have taken the voltage gated calcium channel with the calcium buffering. Voltage gated channel is the different from the sodium calcium exchanger. Calcium ion enters through the voltage gated channel that we have taken in the account in that. Here, to indicate the role of cytosolic calcium concentration, we have used the computation model, and we have included the sodium calcium exchanger into the previous model that we have used previously, calcium buffering in the astrocytes. Another thing we have done here, we have just framed the shape of the astrocytes. Previously, we have taken the smooth geometry, just like the rectangular, just like the circular cells. We have taken the smooth boundaries there, so that we have used the geometry of the astrocytes. Try to find the geometry. This is the calcium buffering. What happens as the calcium enter into the nerve cells? This buffer term that react with the calcium ions and make it the calcium bond buffer. It means what? It reduces the calcium concentration level so that it not become the toxic. Also, it will also block the nerve impulse from go from one cell to another cell. Just like for example, a very good example we see when we feel the pain. What happens? We take the painkiller, and the process occurs at the nerve cells. The particular kind of buffers proteins react with this calcium ions so that it reduces at such level the pain, the neurotransmitter which is responsible for that pain, it will not move from one cell to another cell. That happens in the calcium buffering processes. Using the Fickle diffusion law and the law of mass action, this is the general equation we have taken in the form of reaction diffusion equation. sodium calcium exchanger, how calcium go out from the cytosol. This is the process as 3 sodium ions enter into the cell and it releases 1 calcium ion from the cytosol. In this way, what happens? It decreases the calcium ions, so that the flux of the J flux that is formulated in that way. And the final, what we have the equation, that is the second order. We have taken the second order partial differential equation. The bonding condition we have framed accordingly. This we have taken the shape of the astrocyte. We have tried to form the star shape that varies from the region to region in the brain. It's simple is we have taken the star shapes, and using the FEM and the MATLAB we have tried to find the solution. These parameters, these values we have taken to simulate the results. What we see in the simulation, this we have taken here only the diffusion process. That is, we have taken the absence of both the parameter first, buffer and the sodium calcium exchanger. It means what? It is a simple diffusion, second order diffusion equations, right? Analytically also it has a solution of the negative exponential way that we see that we have taken at here the first boundary condition, and the calcium enters, it goes down gradually, decrease, and finally it maintains the background concentration. As we include in the model, in presence of buffer and the sodium calcium exchanger, what we see the result, the behavior, since we are using the second two-dimensional diffusion equation, it should be the negative exponentially, so that when we include this buffer in NCX, it decreases the calcium concentration level throughout the region. Physiologically, it has the role to reduce the calcium ion level throughout the region. Second is what we see. We have we have taken the absence of the sodium calcium exchanger. We are seeing only the impact of that calcium buffering. In that there was such increase we found due to the absence of sodium calcium exchangers. It has impact of only calcium buffering processes. That we have got the result, and we have used here directly the impact of all these only 2 parameters. We have not considered the association rate, the particular amount of buffer. We have not considered particularly any other parameters, just like the diffusions. We have used simple, because in the literature we have done this calculation. Here we have taken the simple impact of this. It is observed that the effect of buffer and NCX is more significant on the source of the mouth, because the buffer exists on the source also, and the channels also on the plasma membrane it exists. The effect of the NCX is found significant in less amount of buffer. The triangular element used here for the FEM process, this gives the better approximation. The role of NCX is found important to reduce the calcium ion concentration in the astrocytes. And the model developed here makes the use of finite element easier. It is also proposed here we may include the other parameters, physiological parameters, just like the endoplasmic reticulum, the impact of mitochondria, because the buffering process also exists inside the mitochondria and the inside of the endoplasmic reticulum. That may be included with these parameters, and we have the more ideas, clear ideas regarding these processes. These are the references. This is the acknowledgement. I thank our institute for the financial support to carry out this research. Thank you.
Any questions? Okay, no questions. Thank you very much.
Thank you.
So, Mr. President, please.